Welcome to the video on Computer-Aided Drug Delivery System, or CADDS. I am Dr. Krishna Yadav, and I will be guiding you through Unit 1, Part 1 of the MPharm Pharmaceutics curriculum. This part of Unit will cover the history of computers in pharmaceutical research and development.
We are witnessing a paradigm shift from hit and trial to rational design. The old way involved random screening and manual labor. The new way utilizes predictive modeling, AI integration, and automated manufacturing, covering the molecule, the form, and the process. This transition is from using computing for administrative support, like data storage, to it becoming the central nervous system, providing data intelligence. This is a shift from simple processing to ensuring the effective functioning of multi-layered R&D stacks. The strategic victory is compressing the timeline. Convergence of AI-driven design, 3D printing, PBPK modeling, and robotics have helped finalize the shift from serendipity to engineering. This has reduced the R&D timeline from about 20 years to roughly 10 years. The paradigm shift moves from the 1940s administrative utility, using early computers for payroll and accounting, to today's in silico design. This allows for rational, predictive design, making computing the central nervous system of research. The 1960s were the point of origin, shifting from intuition to mathematics. This decade saw computing migrate to corporate R&D and the birth of QSAR. Discovery became a mathematical science, with hardware like IBM mainframes and the introduction of the quantum chemistry program exchange. The 1970s focused on digitizing the library, with hardware costs dropping and the introduction of VAX and 8-inch floppies. Foundational data repositories like the PDB and CSD were created. A collaboration gap emerged, as medicinal chemists struggled to view the computer as a partner. The 1980s marked the rise of CADD, a strategic bet on atom-by-atom design. CADD became an official discipline, but initial failures led to a forced retreat. Key tools included ChemDraw, Amber, Autodoc, and TOPKAT, following the typical hype cycle. The 1990s were the era of automation and screening, focusing on mass testing. High-throughput screening, or HTS, utilized combinatorial chemistry. The focus shifted from rational design to mass testing, with Lipinski's Rule of 5 transforming efficiency. Success stories include dorzolamide and HIV protease inhibitors. Drug design methodologies are divided into structure-based, SBDD, and ligand-based, LBDD. SBDD includes de novo design and virtual screening. LBDD includes QSAR, pharmacophore modeling, and similarity searching. Lipinski's Rule of 5 acts as a filter for oral candidates. CAD has several success stories. Norfloxacin was the first SBDD-based molecule to market. Dorzolamide, in 1995, was the first carbonic anhydrase inhibitor via SBDD. HIV protease inhibitors were a triumph of docking. Vemurafenib, in 2011, was the first fragment-based drug design drug. The 2000s ushered in the genomic era and big data, focusing on omics and virtual screening. The Human Genome Project was completed in 2003. The shift was from single genes to omics. New tools included routine virtual screening and PBPK modeling like GastroPlus. The 2010s to the present is the intelligence era of AI and deep learning. GPUs and TPUs enable massive parallel processing. Deep neural networks can predict toxicity and bioactivity better than humans, reducing the discovery and optimization timeline significantly. The transition to computer-aided pharmaceutics moves from molecule to medicine. Discovery involves CADD. Formulation uses pharmaceutics to model solubility and stability. Delivery simulates release and absorption. The crucial step is making the medicine once the drug is found. We see the eras of experimental design in engineering quality. From agricultural origins to the present quality by design QBD era, the strategic impact has shifted focus from final testing to building quality into the design space through science-based definitions. Codifying expertise through rule-based logic and expert systems mimics human experts using if-then rules. Case studies like Formulagic and the CADDS system demonstrate reduced development time and suggested formulation conditions. The 3D printing revolution, or additive manufacturing, progresses from prototyping to direct manufacturing. Key milestones include stereolithography in 1984 and the first FDA-approved 3D printed drug, Spritam, in 2015. This allows for functional end-use parts. The hardware origins of additive manufacturing started with stereolithography, or SLA, in 1984. It advanced through selective laser sintering, fused deposition modeling, and eventually binder jetting, which is the ancestor of pharmaceutical printing. 3D printing is shaping medicine. The zip dose process involves powder spreading, liquid binding, and layering. The 2015 approval of Spritam, levetiracetam, highlights the benefit of personalized dosages and rapid disintegration. Spritam uses zip dose technology, involving a powder layer, fluid deposit, and interaction to create a highly porous structure. Successive layers are printed up to 40 times, resulting in a tablet that disintegrates instantly, unlike traditional compression.
Depicting the expert system and neural computing, this illustrates a workflow for how artificial intelligence, specifically expert systems and neural networks, is applied to pharmaceutical drug formulation. It outlines a computer-aided approach to designing and predicting the success of medications. This further shifts the focus from the design of the drug itself to how the human body reacts to it, a field known as pharmacokinetics, PK. While the previous diagram was about formulating the physical tablet, this one is about simulating the drug's journey once it is swallowed. A fantastic companion piece to the previous one focused on the general concept of simulating the body, ADME. This one acts as a specific historical timeline, showing exactly which software programs made those simulations possible over the decades. This timeline tracks the evolution of industrial robotics, showing the shift from heavy, caged machines in the 1950s to today's safe, collaborative robots. Nature is now inspiring the future of medicine, with soft, biomimicking robots designed to safely navigate the human body and deliver treatments precisely where they are needed. By shifting from rigid metals to soft materials like silicone, scientists are creating micro-robots that mimic caterpillars and octopuses to revolutionize targeted drug delivery. Early knowledge-based systems transformed pharmaceutical development by replacing human guesswork with structured logic, helping major companies significantly cut down their formulation time. Before modern AI, these early digital brains used decision trees to solve complex formulation problems, reducing drug development time by up to 35%. By utilizing neural networks and virtual stress testing, scientists can accurately predict both the chemical success and the physical flaws of a drug before a single pill is actually manufactured. Advanced AI and in silico simulations allow researchers to solve complex formulation problems, moving beyond basic statistics to predict everything from molecular efficacy to physical tablet cracking. To prove a drug is actually safe and effective, researchers rely on a validation layer, an evolving ecosystem of PK/PD software that ranges from early population modeling tools to today's open-source platforms. This timeline breaks down the gold standard software tools like NONMEM and GastroPlus that the pharmaceutical industry has used over the decades to mathematically validate drug safety.
Challenges remain in scaling up production, standardizing materials, and meeting stringent regulatory requirements for consistent quality.
Today, deep learning acts as the central hub of modern medicine, combining massive datasets and genomics to create highly targeted precision treatments. By transitioning to advanced deep neural networks, scientists can now process immense biological complexity, leading to breakthroughs like the very first fully AI-generated drugs. This evolutionary map shows how decades of breakthroughs in computing power, from early mainframes to modern GPUs, have directly fueled the advancements in both drug discovery and pharmaceutical manufacturing. Tracking the history of pharmatech, we can see how the parallel tracks of computing, chemical discovery, and robotic production have continuously influenced each other, ultimately converging in today's era of deep learning and 3D printed medicines. Here are the acronyms and their description used in this video. Here are a few quoted literatures and source for further studies. Thank you for watching this video. We hope this content provided a clear understanding. Please like, share, and subscribe to my channel.